Okay, excuse me. All right, I'm doing this with a bit of a head cold, so I hope this is understandable. Um, I want to talk just a bit about how speciation happens and how we can see its effects and how fast it happens. What's the tempo? And probably the most awesome place where you can see the effects of a lot of speciation are what we call adaptive radiation. And in an adaptive radiation, you have a founder species encountering a habitat or probably a group of habitats with a lot of unoccupied ecological niche space and speciating relatively rapidly into a whole bunch of closely related species. Uh, that whole bunch of closely related species we call a species flock. If you drew a cladogram, it would look almost like a broom with lots of species radiating from that common ancestor in a short interval of time. Some classic examples of this would be the Galapagos finches are probably the most famous. There's 14 species of finch in that small cluster of volcanic islands. The Hawaiian honey creepers, I think there's 58 species of those, although about half of them have gone extinct in recent times, unfortunately. But as I often say, if the beagle had gone off course and ended up in Hawaii, Darwin would have written very much the same book, except it would be about, we'd be talking about Darwin's honey creepers, not Darwin's finches. Over 100 unique species of French Polynesian land snails. Another great example. Unfortunately, many of those have gone extinct in recent years. The over 700 species of fruit flies, genus Drosophila, and a related genus called Scaptomyza in the Hawaiian Islands. And about 1,000 species, unfortunately, many of which have gone extinct recently, of African Rift Valley cichlids, cichlids being a type of fish. We're not going to look at all of these, we'll look at some. You've already seen a bit about the Galapagos finches. The Hawaiian honey creepers, um, that's a family that's undergone an adaptive radiation into 57 species. I think the previous slide said 58. There may be some debate on the matter. Unfortunately, 39 are extinct or have probably gone extinct. Um, Fortunately, I think we still have all of the Galapagos finches. Um, Species flocks like this can be at risk of extinction because each species may occupy only a very restricted region. They're not widespread. They may be very dependent on one particular food source or something like that. Um, so they tend, if anything affects that one small area where a species lives, or the one food it's specialized to feed on, or something like that, it may go extinct quite quickly. Little problem for conservation right there. But anyway, the honey creepers parallel the Galapagos finches in a lot of ways. Um, Typically, they feed on native flowers' nectar, hence the name honey creepers. But there's some that feed on insects or crack seeds or feed on fruit. And as you might expect, they've got different beak types that show adaptation to the lifestyle of each species in much the same way that the Galapagos finches have the same kinds of adaptations in their beaks. So here's a few. The Vestiaria coccinea up there at the top, the iiwi, that one is still with us. That's a nectar sipping bird, and it's got this very long curved beak, very good for slipping into the corolla of a flower and slurping up the nectar. You can see a very long, narrow beak for, I think, much the same function in the Amakihi up at the top left. Sitarostra has this big parrot-like beak. Chlorodops has a big crunching beak that's clearly a seed eater right there down at the lower right. Sitarostra, I figure, would be a seed and fruit eater, the OU. And so, again, you've seen this before, the way you can have a cluster of related species that nonetheless differ in the ways they're adapted to their environment and specifically their diet. And the Hawaiian, like the Galapagos, the Hawaiian Islands haven't been around for that long. We'll talk a bit later as to exactly where they come from because it dovetails very nicely with the evolution of Hawaiian species, as we'll see. But the Hawaiian Islands are not particularly old. In fact, the one of the volcanoes on the Big Island of Hawaii has been erupting on and off pretty steadily since 1981. And so that island is still being built. Um, and so, in a relatively short length of time, just a few million years, which doesn't sound short, but it is compared to the total time that the Earth has been around. In what is, believe it or not, a very short length of time, we have seen quite a lot of speciation taking place in the Hawaiian Islands. Another classic example is the African Rift Valley cichlids. The East African Rift Valley is actually a place where the African continent is very, very slowly pulling apart. Um, we still have several million years to go, but it cuts southwards more or less from Ethiopia all the way down to Mozambique. And eventually, Africa will break apart, the ocean will rush in, and East Africa will end up as a very large island, similar to Madagascar, which broke away from the African continent many millions of years ago. Anyway, when a continent splits apart, along the crack, a deep valley tends to develop called a rift valley. And so the East African Rift Valley cuts from Ethiopia all the way down to Malawi. And in some parts of that Rift Valley, some parts are desert, but in others, there's some large lakes that have formed. Incidentally, there's something similar close to us in North America. The North American continent started rifting apart approximately 180 million years ago. It didn't end up cracking all the way through, but we've got this deeply buried crack in North America called the Real Foot Rift that cuts up right through the south central United States. One little branch of that is known as the New Madrid Fault. You might recall that earthquakes on the New Madrid Fault produced, what was it, an 8.6, the most devastating earthquake ever recorded in the continental United States. Well, it wasn't super devastating because there weren't many people living out here because it was in 1811. But if it happened today, say goodbye to Memphis. The New Madrid quake did things like ring church bells in Connecticut, cracked sidewalks in South Carolina, if memory serves. And it happened because we're sitting on top of a very ancient rift valley that you can't see on the surface because it's been filled in with eroded sediments from the Appalachians and the Ozarks and the Ouachitas, which used to be a lot higher than they currently are. That rift has been filled in, and we now call it the Lower Mississippi River Valley. but we've got a big rift buried deep under all of that. So next time you're driving, round about Palestine, Arkansas, there's a great big rift under your feet. So there. Anyway, there's a rift that's still exposed on the surface in East Africa, and the lakes that fill up the East African Rift Valley contain species of cichlid fish. Lake Victoria used to contain 300 species. Unfortunately, humans introduced the Nile perch, um, which is a predator. It's not native, and it ate the cichlids like popcorn and has probably wiped out about 200 of these species. The other lakes in the East African Rift, especially Lake Tanganyika and Lake Malawi, each of those contains a similar flock of cichlid species. And there are, or were until recently, about 1,000 species of fish in all of the East African lakes. A few of these are actually popular in the aquarium trade. If you any pet store around here with a good aquarium department will probably have a tank of African cichlids. They're quite colorful behaviorally. They're quite interesting. Um, and, people who like caring for fish sometimes like taking care of African cichlids. So you can, so you can go down and see some in PetSmart on any given day. And they're all different from each other in color, of course, in size and shape. Some of them have very complex breeding behaviors. Some of them, for example, are mouth brooders, which means one of the parents, I think it's often the male, keeps the eggs in his mouth until they are mature, until they hatch and the young are mature enough to leave it. Different sources of food. There's okay, there's East Africa. And the Rift Valley more or less does this. And there's Lake Victoria in the middle of it. And Lake Tanganyika on the border between Tanzania and the Republic of the Congo. And there's Lake Malawi between Tanzania, Malawi, and Mozambique. So that's where the lakes are that we've been talking about. And the next slide shows 6 of the 300 plus cichlids native to Lake Victoria alone. So these are 6 that the Nile perch didn't quite get. Some of these are males in breeding coloration. Males develop what are called egg spots down here on their lower fins. But you can certainly see they're different colored. If you look a little more carefully, you can see difference in things like mouth size and mouth angle. Like Pixochromis right here looks like it's got a pretty big mouth, and Aureochromis looks a bit smaller. That is probably correlated with prey type. Some of these cichlids would be insect feeders. Some of them are snail crushers. You wouldn't see this externally, but the snail crushers tend to have these teeth in the throat that grind together and break up snail shells. Others are probably insect feeders. So they occupy a number of different niches. And of course, they're different in mating behavior. A lot of them have pretty sophisticated mating and courtship and mating behaviors. They're all different. And the thing is, we know that Lake Victoria dried up completely 12,400 years ago. We know that because if you bore a hole into the lake bottom and bring up a sample of the sediments, under the silt of the lake, there's a layer of soil. So that means those 300 species of cichlids must have descended from a common ancestor in less than 12,000 years. There's a small lake called Lake Nabugabo that's been separate from Lake Victoria for only 4,000 years, and yet there are 5 cichlid species in it that are not found anywhere else in the world. This may be an example of what we call peripatric speciation. This is not really different from allopatric. Peripatric refers to speciation on the border, on the fringe, on the perimeter. If you have a deep lake, imagine it dries up for a bit. Excuse me. Imagine you have a lake and it partially dries up. Unless that bottom is really smooth, it's not going to just all dry up at once. The lake will shrink somewhat, but there will if the lake has kind of a bumpy bottom, there will be little ponds and puddles left around the outside of the lake. And the idea is that if the lake level keeps fluctuating up and down, when the lake dries up, you'll have cichlid populations, fish populations that are cut off from each other in these little ponds on the edges of the central lake. And then if the lake fills up again, those ponds will merge and all of the species will come back together. So the idea is that that may be why we've got so many fish in Lake Victoria, or did before the Nile perch ate them like popcorn. Lake Tanganyika's held water for 3.5 million years, and the cichlids are even more diverse in shape than Lake Victoria's. So you can see, if you look carefully at the scale bars, we've got very different sizes, different mouth shapes, and again, that's probably correlated with diet, different builds, correlated with swimming ability, and so on. And it's probably a very similar story in Lake Tanganyika. There's just been more time for speciation to happen. Here's the thing. I've just mentioned that we've had this burst of speciation in Lake Victoria in the past 12,000 years. For reference, by the way, 12,000 years ago, we still don't really have civilizations yet, but there were fully modern people almost everywhere on the planet by that time. That's roughly the time in which the earliest Native Americans are starting to come in. There were people in Europe, people in Asia, certainly people in Africa, and they were fully modern. They were indistinguishable from us. So yeah, written history doesn't start until just a bit later once people have figured out writing. But that is well within the lifespan of our own species. The old assumption as to how speciation happened was that species had to change gradually, except in cases of speciation by hybridization and polyploidy, which was often ignored because a lot of the early figures in modern synthesis evolution were specialists on animals, not on plants. And for some reason, plants get up to silly chromosome tricks more often than animals do. Oh, a guy named Richard Goldschmidt, um, brilliant German biologist. Poor guy. In Germany, he had a big lab and a big staff of research assistants. He was one of the most respected biologists at the time. He ran afoul of the Nazis, managed to escape, made it to the University of California at Berkeley. And unfortunately, the only job they could find for him was teaching freshman biology. So this poor guy who, had been a high-powered researcher and who didn't speak English suddenly had to get up and lecture to a bunch of freshmen. He did survive, but I gather he was rather inconvenienced for a while. Anyway, we'll look at his ideas in a little bit more detail coming up next week. He'd suggested that you could get new species and maybe even new genera, new orders, new classes that might arise by macro mutations. That's mutations that have a major phenotypic effect on development, mutations that change the phenotype in a major way, not just the color of the flowers or the shape of the pea pods or what have you, but that reorganize the phenotype in a much larger way. Um, it's a revival of, if you remember, Thomas Henry Huxley had argued for saltationism, the idea that evolution could make jumps. So this is kind of a saltationist argument. It derisively, this was a joke, it became known as the hopeful monster theory. The idea is that a mutation that radically alters the phenotype produces a monster, but hopefully one of those monsters might be adapted to the environment and survive and propagate its kind. Goldschmidt probably wasn't right, but he was onto something. And next week, we'll look at what he was onto when we can talk about developmental biology and how that applies to evolution. The old assumption was that directional trends within a lineage are dominant. That if you looked at species A, every 50 years over the course of a million years, after 50 years, it would look a tiny bit different. And after 100, a tiny bit more. And after 150, a tiny bit more. And so on. You get this gradual shift in phenotype. And that directional trend within a lineage is called anagenesis. And if we don't see this in the fossil record, it's because the fossil record's not perfect. It is perfectly true, by the way, that the fossil record is not perfect, because we don't have anything like the total number of fossils that have ever been produced. Nowhere close. We're still finding new species at a steady clip, and many once living things just haven't left any fossils at all, either because they're squishy and would not normally fossilize, or just because metamorphic change to the rock layers or erosion or something like that has obliterated their fossils. Jim Valentine estimated that if you took a typical habitat in the shallow ocean, only about 13% of the invertebrates would be expected to leave any recognizable fossils at all. The other 5, 6 of the critters that are there are squishy or delicate and would not normally leave any fossil trace. So we know we're not seeing everything when we look at the fossil record, but this is sort of the early model of evolutionary change. You start where a population has some trait that has a normal distribution. In other words, it's our old friend the bell curve. And that bell curve shifts in a steady direction in one direction over time. That would be anagenesis. So imagine a bell curve very slowly sliding to the right. The same kind of pattern that we saw when talking about directional selection. Excuse me. Push that bell curve over in one direction for long enough, and eventually you'll get something that's so different, we call it a new species. So that was the old model, the model of anagenesis. And this was kind of assumed when looking at fossils like this. This is a snail from rocks about 50 million years old in Texas called Athleta petrosa. And you'll notice that there's a trend from older to younger specimens to get larger and also to get spikier. So Athleta petrosa smithi right here only has little spines. Athleta petrosa symmetrica has much bigger ones, and the overall size of the shell is bigger. So that's what people thought evolution looked like. You have one form that gradually shifts into another form and another form and another. That's anagenesis. There's some actual data that seems to show that. What's been done here is scientists took samples of the shells of a single-celled protist called Lepidolina. There are single-celled organisms that make shells, lots of them. This is a type of, it's called a fusolinid foraminiferan. Take invert with me and I'll talk a lot more about foraminifera. Let's just say for right now, it's a giant amoeba that's got a shell. And what they did was for every sample that they took, measure the diameter of a portion of the shell called the proloculus, and estimate, calculate the mean and estimate the standard deviation, and see how that shifts over time. And you can see there's kind of a trend here. It might zigzag a little bit, but the overall trend has been for this population of protists to get larger. The mean's gone from 200 microns to nearly 600 microns. The variation has also gotten quite a bit wider, and that's change over about 15 million years. So that's what anagenesis would look like. Enter Stephen Jay Gould, paleontologist who specializes on land snails, and Niles Eldridge, who specializes in trilobites, in the 1970s. They argued that if you honestly look at what the fossils tell you about how species form, you don't really see that kind of slow, gradual shift. What you really get is species tend to stay the same for millions of years. They might zig and zag a little bit, but they don't significantly change. And new species appear relatively abruptly. Now, this gets confused a lot with Richard Goldschmidt's theory that new species can appear in one generation. What I'm talking about here, the species don't appear in one generation. What happens is the time that it takes for new species to form is a very small fraction of the time that those species will persist. That when you get the evolution of a new species, it takes place maybe in just a few thousand to tens of thousands of years compared to the millions of years that that species might persist. The length of time when you get change is small relative to the total length of time the species is going to be around. And the name they gave for this pattern is punctuated equilibrium. Or if you don't feel like saying punctuated equilibrium, it's punk-eq. That's not a theory of how species form, it's a description of the pattern of species formation over time. Equilibrium refers to the fact that species tend to stay the same for fairly long periods, punctuated by episodes of relatively rapid change. Okay, so what's this look like? You know what? I'm going to skip this slide. I used to share well, we were never office mates, but I was across the hall from this guy back in the 90s, a grad student named Ross Name, who's since gone on to, I believe, City University of New York. And he demonstrated evolutionary transitions in a little group of marine snails called marginellids. These are a couple of marginellids. They're not very big. You'll sometimes see them used to make shell jewelry, but other than that, they don't have a significant commercial use. The common name, when they're called anything at all, they're called coffee bean snails. So there's you a coffee bean snail. And Name had access to a very big collection of marginellids from Costa Rica, from fossils, and he made a whole bunch of measurements on them. This diagram from his paper shows the measurements that he made, and he used a technique called principal components analysis to combine all of those measurements into a single number. The you can't plot all of those measurements at once because it's hard to find 10-dimensional graph paper, right? You can't keep track of 10 variables at once. So principal components analysis is a way that you can take all of these numbers and boil them down into one or two that express the largest possible fraction of the variance in your sample. So when he did this thing down here at the bottom called discriminant axis, that's the same as his first principal component. It's just a number that combines size and shape. And so shells with a high value tend to be kind of broad, and shells with a low value tend to be kind of narrow and thin. And down here at the lowest level, he basically got a bell curve. So at levels A, B, C, and D, not a huge amount of change in the average phenotype. And then from D to E, things get very different. And by the time he's up here at level F, we pretty much got a split between the wider Prunum coniforme, which is over here, and the narrow, slender Prunum cristae right there. And you can see that by level H, there's still a few coniforme that are still around. Christine Laddie seems to be dominant. And you can see that most of that change takes place only in one interval right here, right? Let me change my writing tools. From A to D, there's not a humongous amount of change. From F to H, there's not a huge amount of change, and it's only in E that we get that major burst of change. So there is a gradual transformation, but what I want you to see is that the E interval, this time of transformation, takes up a relatively small length of time with respect to the total length of time that we're talking about. If you look, E is those rocks right there that I'm shading in if you look at the diagram over on the left. And so we go from rocks layers A, B, C, all the way up to D here, where we have Prunum coniforme. We catch that one little interval right there where the change happens, up in rock layer E. And then F through H is dominated by Prunum cristae. So the length of time where that change happened right there is a relatively small fraction of the total length of time that we've got. Okay, hopefully that's clear. All right. Last thing I want to introduce, I'm not sure what time it is. I can't see my my phone, but let me just finish this and give you guys and myself a break. Hi, cat. Sorry, my cat is informing me that she has been insufficiently fed. Hello, you annoying beast. Okay, there's a whole group of single-celled eukaryotes called foraminifera. The bodies are vaguely amoeba-ish. We don't really need to go into that. But what forams do is secrete a shell-like covering called a test. The word has nothing to do with an exam, by the way. Testa was Latin for a pot. And you'll often see it used to refer to a structure that reminded somebody who was probably on drugs of a pot. So this is a foram test in the picture over at the left. Typically, it's made of calcium carbonate, meaning it's kind of chalky, or it's agglutinated, meaning that the cell picked up tiny sediment grains and glued them together. And that test is shot full of little holes called the foramina. The reason these are useful is that forams are stupidly abundant in the ocean. There are places in the deep ocean where the sediment is almost entirely foram ooze, the skeletons of forams that have died and sunk to the bottom. There are bodies of rock that are made up of almost nothing but foram tests. And the upshot is that if you drill a core of deep sea sediments, you can divide it very finely and you can trace evolutionary changes in the forams in very fine detail because you've got many thousands of tests, each of which you can measure. And your record records very narrow time intervals. It's a fine grained record of change. So when we do this, what do we get? These shapes right there are like that. That's the outline of a foram species that happens to be called Globorotalia tumida. Oh, look, I'm coloring. There's a Globorotalia tumida. You're just looking at the outline of the test. Hi, cat. Yes, I've got you. I don't know if you can see this, but I'm cuddling a cat who doesn't really want to be cuddled. Get out of here. You want to go? Poor kitty. I think I just got cussed out. Right. If you look on the y-axis, that's time. And between about 10.4 million years ago and 5.5 million years ago, that's 5 million years in this one zone of, I think, the Atlantic, we have a species called Globorotalia plesiotumida. On the x-axis, you can see that what's being measured is the cross-sectional area in square millimeters. And between about 10.5 and 5.5 million years, there's not a great deal of change. There's little bits of zigs and zags, but there's mostly things are pretty stable. Between about 5.5 and 5 million years ago, the species does a zigzag. And then after about 5 million years ago, it still zigs and zags a little bit, but we have what's considered a new species called Globorotalia tumida. So the idea is that this is an example of punctuated equilibrium. There's only about 500,000 years where you get this major episode of change. For about 5 million years, we have Globorotalia plesiotumida, which doesn't really change very much. After about 5 million years, we have Globorotalia tumida, which does change a bit and zigzag, but not enough that we call it separate species. And then it's only in this little interval Right here, I think I'm having a little bit too much fun with these pens, but it's only in about between 5.5 and 5 million years ago that we see most of the change from plesiotumida to tumida. That is punctuated equilibrium for you. Um, Equilibrium means that species phenotypes tend to stay the same, like there and there, punctuated by relatively rapid bursts of change. Now, I've seen people try to use punctuated equilibrium as a refutation of evolution. Most people now seem to agree that really, what's going on is consistent with evolution as we understand it, because evolutionary change tends to be most rapid in small populations. Small populations have more genetic drift. For small populations, it takes less time for new mutant alleles to become more common. Small populations, maybe, in your typical allopatric model, may be responding to selection, and they can respond faster. Small populations are where the change is going to be concentrated. Once a population is large, just being big tends to slow down the rate of evolutionary change because it's got much less genetic drift, and it will take much longer for mutant alleles to become common, if they ever do become common, which they may not, once they appear. So your greatest rates of change is going to be in the smaller populations. Big ones tend to be buffered from evolutionary change. They will tend to stay where they are. Mm. And that's perfectly consistent with punctuated equilibrium. If you've got a very large population, we would not expect to see that population um, change. We would not expect to see it gradually shift. It's going to be the little ones, the little isolates in the ponds around the edge of the lake, where the change is going to be concentrated. And there it can happen pretty fast. We've seen cases of speciation taking only a few thousand years. I've mentioned several over the past couple of lectures. Now, from the point of view of somebody who works on modern organisms, let's say it takes 10,000 years for a new species to form. 10,000 years is unimaginably long. 10,000 years ago, we were all still hunter gatherers. No cities, none of what we call civilization. From the point of view of a paleontologist, 10,000 years ago is such a short time that you can't even measure it very well with the dating tools that we have on hand. In most cases, using the rocks to try to detect a change over 10,000 years would be like trying to time a track meet with a calendar, right? Calendars are useful for measuring time, but, a 100-yard dash takes place in a much shorter interval of time than a calendar is good for measuring. And it's the same kind of thing here. That transition from one species to the next may be so rapid that we can't always detect it in the fossil record. With the forams, we can, because that's a very rich fossil record. But for many of the shells you would just pick up in the Ozarks, we may not be able to detect it at all. So whether an evolutionary pattern looks like it's gradual or it's punctuated, that might depend on your observer's choice of time scale. And anagenesis is what we popularly think of as the entirety of evolution. There's that famous March of Progress diagram, with Bonzo the chimp at one end and Fred the naked white guy at the other. My major professor actually had a student who asked whether human ancestors had genitals. He reassured her that they did, but she'd only seen diagrams like this, where everybody's legs are turned in such a way that you can't see their junk. In case anyone was unclear on the fact, yes, we believe that human ancestors did have junk. Right. Anyway, that implies anagenesis, where everybody progressively gets slightly taller, slightly walking more upright, slightly less stooped, slightly bigger brains, until we go up to modern Homo sapiens right there at the front, marching off into the future with bold strides. That's what people often think evolution is about. And of course, it's been parodied ever since. So that's one that's been going around for a while, showing our evolution and then our de-evolution as we crouch before our computers. Supertramp was this British group in the 70s and 80s, and that's one of their album covers. But what predominates far and away above anagenesis is cladogenesis, branching. Evolution makes trees. This is still prone to some revision, but this diagram gives you a better idea. Each one of these dark blue bars is the length of time when a taxon has been around, and we infer that these taxa have, let me change the colors, that these taxa have branched and branched again. In a pattern that looks like a big, sprawling, messy cladogram. And ultimately there's us right there. Evolution makes trees. This is something I feel like people don't understand enough. That evolution doesn't really look like that march of progress where each model gets replaced by an upgraded model, making, humanity, I don't know, the iPhone 6 of evolution or something, or whatever number they're up to. Evolution looks like this. Evolution branches. There, added a few branches right there. Evolution makes trees. Okay, that is a good place to pause. So I'm going to stop the recording.